Today's topic is hydrolysis of protein and separation of amino acids. So if you look at a protein, for example, protein is like this. This is a amino acid. And when it progresses like this is the R group attached. And then this is NH, is CH, and this is R, C double bond O. Now look at this. This is the protein. And if we continue like this with another amino acid like this, so this is a protein. Let's say, for the sake of argument, I'm going to use the bio names just to make you understand. Let's say these R groups, 1, 2, 3. If you keep changing these R groups, for example, this is glycine, this is ASP, this is LYS, lysine. Now these three amino acids, which I can see combined together to make a protein, I assume that these amino acids are glycine, ASP, and lysine. Similarly, if I continue with them, for example, again there is a glycine, there is a lysine, and so on. So this is a long chain that's known as polypeptide or protein. So when protein or polypeptide undergo hydrolysis, they react with either dilute acid and heat, or they reacted with the dilute alkali and heat. And when they react with the dilute acid, initially they don't break into amino acids. They break into this glycine, ASP, and lysine, which is known as tripeptide. Okay? And similarly, the bond breaks from here. So the other side is what? Glycine and LYS. This is known as dipeptide. So a protein which consists of the long carbon chain, they break down into the units of 3 and 2, right? And form what? Peptides, dipeptides and tripeptides, which further undergo hydrolysis. To form what? Amino acids. Now these glycine separated from ASP, separated from LYS. Now in a beaker, they got separated. Now this is what? This is mixture of amino acids. Similarly, it undergoes a further hydrolysis to form glycine plus LYS. This is the mixture of amino. So I hope you got my point. What I'm trying to say, that the proteins, when they undergo hydrolysis, initially they turn into the smaller units known as tripeptide and dipeptides, and these dipeptides further hydrolyze under the same conditions, acidic or alkaline, to form mixture of amino acids. And when you write a protein from a tripeptide, the difference you need to understand, because they ask you to calculate the MR, so you can understand the difference. For example, if I change these R group, for example, the first R group is hydrogen. The second R group, let's say it is CH2COOH. And the third R group is CH2, 4 times NH2. Okay? So these are, now this is protein. But if I want to write a tripeptide now, I can't show that the bond length on the opposite side, this one and this one, is still progressing. I have to stop it. So what I have to put it? I have to put OH here. Okay. And similarly, I have to put what? H here. So please mind this difference. If you show the tripeptide, you don't have to show the length of bond is increasing. You just have to show the three carbon chain. Okay. 1, 2, 4, 2. Right. So this is tripeptide. So please draw the structure of tripeptide here. This is a tripeptide. और ये कौन सा tripeptide है? I would like you to draw this, please. This is tripeptide consists of what? Glycine, ASP, and lysine. Okay, so it's a tripeptide. So I hope you understand. Tripeptide is the one which is not further progressing. Only the dipeptide, only the protein is further progressing. Okay. Here it's a glycine, ASP, and this is LYS, lysine. Right? Now, can you please draw the dipeptide of these two? Draw the structure, please, everyone. Draw the structure of glycine and lysine dipeptide. Glycine amino acid and lysine amino acid are shown on the top. Glycine and this is lysine. Yeah, it further breaks down to give amino acids. No? You see? No, they only hydrolyze. Aris, can you show the dipeptide, the structure of dipeptide showing glycine and lysine amino acids? Okay. Here is the glycine and here is the lysine. Just show the dipeptide showing these two amino acids. So in dipeptides, you do not extend the chain. This is H, the first one, glycine. And then here the second one, H, CH2 4 time, NH2, and then COOH. So this is what? This is a dipeptide. So again and again, that you are not supposed to extend the bonds. If I remove the OH and H, these OH and H, these two, and I show it like this, that won't be a dipeptide. It's a polypeptide. That would become protein structure. Okay, so once they ask you to draw the dipeptide, you have to draw like this. And they ask you to draw the polypeptide, so you have to draw the. Okay, now move on. So what I said, the protein is a polypeptide consisting of many amino acids connected together to make a long chain. When you undergo the hydrolysis, first time it breaks down to the smaller units, dipeptide, tripeptides, which are further hydrolysis, undergo the hydrolysis to form the amino acids. Okay. And this mixture of amino acid is then present in the beaker, and then they undergo the hydrolysis. Then they need to be separated. Now heading the separation of amino acid. Yes. मेरे पास फिर होता है कौन से पेज पे तो दूसरा कौन सा ओ लेवल का फिर कोड है 5070. Okay, separation of amino acids, please. Now, please pay attention. This is the mixture of amino acid, bacho. Let's say this is the mixture of amino acid which we have to separate. The today's task is that I have this beaker consisting of 3 amino acids. Okay. Which one? These glycine, ASP, and glycine. Okay, please pay attention. Now the point is that I have to separate them. What technique we apply? Please pay attention. First, listen to me. Then I'll tell you how we get. I'll make you write everything. I'll take a spot from this mixture and put it here. This is a glass plate that has a polyamide gel posted on it, just like a वो नहीं जो हम use करते bio में slide. It's a slide. What I took the mixture of amino acid here. Okay, and before putting it here, I buffered it at some specific pH, and that I suppose 7. This mixture of amino acid buffered at pH 7. Buffered at pH 7. And this mixture contains which 3 amino acids? Glycine, ASP, and lysine. My point is, my intention is to get them separated. This side is positive. This side is what? Negative. How? Because I connected this plate with the negative terminal of the battery on this side, positive terminal of the battery on this side. Right? So what I did, I took a battery connected with the positive and negative terminal of this plate on the both sides. Actually, I wanted to apply a potential difference. So what happens, let's say, I assume at this moment, I'll explain in later how I decided this. Let's say the lysine in this mixture has a positive charge. ASP in this mixture has a negative charge, and glycine is 0. So as soon as I apply the potential difference on this plate, what happens? The lysine is going to move in which direction? Negative. The ASP is going to move in which direction? Positive. And where the glycine is going to go? Got it? So it's not going to go anywhere. So after some time, the same plate will look like this. One amino acid reached at this point, one reached at this point, and one stayed here. I don't wait them to reach to the respective electrodes, the positive and negative, then they will get discharged. I will stop right here, and I'll see the ASP here, I'll see LYS here, and I'll see glycine here, and this way they are separate. So it's a very, very similar, just like electrolysis you did. You applied a potential difference on the mixture of amino acid. One goes in, the positive one goes to the negative side, and the negative one goes to the positive side, and in this way they are separated. इसके ऊपर negative charge था, इसके ऊपर positive charge था, इसके ऊपर कोई charge नहीं था. So I think it's a straightforward thing, right? Is it clear? 2-2. So I hope you got it what I said. So every amino acid may have some charge. So once you apply the potential difference, they go to the respective electrode. Now let's see why they have charge and what kind of charge they have, right? Then I'll make you write all these steps. Look at this. Let's look at the glycine first of all. Look at this, bacho. This is glycine. When you dissolve glycine in water, the other day we said we understood what is the isoelectric point. Let's say pH 7. This is not the right, but I'm assuming at this moment, just to make you understand a concept, कि glycine का जो isoelectric point है वो pH 7 है. When pH 7 is isoelectric point, at least that means the glycine will be what ion? Zwitter ion at this. So therefore, you notice that glycine being a Zwitter ion in this mixture cannot move in any direction. It remains in the center because it doesn't have any charge. It's a Zwitter ion. Zwitter ion, the one which side, one side is negative and one side is positive, and overall charge is Zero. There's no overall charge, right? Similarly, if you look at this NH2, CH2, sorry, this is another amino acid, NH2, CH, CH2, COOH, and then COOH. What is this? It is ASP. When I put it in water at pH 7, let's say, it ionizes to give, look, this H positive, and this nitrogen is going to gain hydrogen. So at pH 7, which is not its isoelectric point, it is going to be NH3 positive, CHCOO negative, CH2COO negative. So Aries, what will be the overall charge at this point? What will be the overall charge at this point? Sorry? Negative. Yes. Why negative? Overall charge is negative because this is one positive, this is one negative, and this is what? They cancel out, so overall one negative. Similarly, if you look at this NH2CH, COOH, and then CH2 4 times NH2. This is a lysine. If you put the lysine in water, pH 7 is again, it's not zwitterine. It's isoelectric point. So it's going to be what? NH3 positive, CHCOO negative, and then CH2 4 times NH3 positive. Okay? Okay? So now what is the overall charge, Aris? Yeah, positive, negative cancel out. So what is left? One positive. So that's why the lysine is the positive at H positive. At 7, this is negative and this is what? That the lysine move towards which electrode? The cathode, and ASP move towards anode and glycine towards this. I hope it's clear now. That's why these amino acids move to these respective electrodes, right? Now my next question is, this mixture was buffered at what pH, bacho? Let's say we buffer it at a different pH. For example, now please pay attention. Let's say I buffer it at pH 10. pH 10 mean I have to add what? Hydroxide ions. So hydroxide ions are going to neutralize which side of the zwitterine? No, NH3 positive. So it will turn into what? NH2, CH2, and COO negative. So at pH 10, I hope you understood, at pH 10, the same glycine is not going to be a zwitterine. It is going to be a anion, negative ion. And when you apply the potential difference, it's going to go to the positive plate, the anode, not going to stay in the center. Similarly, if I would have buffered at pH like 5, or let's say 2, so I have to add what? H plus. The H plus is going to attach with COO. So it's going to be COOH, CH2, and then NH3 positive. Okay. So what is the overall charge? So in this case, at pH 2, it's going to move to the right. Okay. Now my question to you: if what will be the structure of ASP at pH 10 and at pH 2? Please give it a shot. Similarly, what will be the structure of lysine at pH 10 and at pH 2? I can tell you pH 10 mean OH add किया है, pH 2 mean H positive add किया है.
सर इस एक COO नेगेटिव पे अटैक करेगा ना H प्लस, एक CO पे अटैक करेगा H प्लस, ये दोनों पे करेगा, एक पे करेगा.
Yeah. Haris, can you help me? What is the structure of ASP at pH 10? NH2 बन जाएगा, so rest of the two things remain the same. So it's going to be what charge? Yes, it's going to be 2 negative. What about you? OH neutralizes the H positive ion. Na? H3 positive, mein there is a H positive ion. NH3 positive, COOH, and it is added in too much excess, so it's gonna add to the both these sides. Okay, so it's going to be what? Positive side. The H positive ions, a lot of H positive ion added, so they're gonna plug all the. It is added in excess.
वो question में mentioned होते हैं.
Yeah. Right.
वो pH से judge करना.
Yes, pH से, or they tell you. Now next one. Look at the pH 10. What about this? You added what? OH negative ion. So NH2 on the both side, na, it is added in too much excess. Okay. So बन जाएगा CH2 4 NH2. So what is this? 1 negative. What about the other one? Yes. So NH3 positive, CHCOO negative, CH2 4 time, NH3 positive. So sorry, yeah, COOH बन जाएगा. So what is this? 2 positive. Do you get it? So these are their changes. Now if I ask you, listen, if I ask you, similar thing, a screen where we can see all of them. At pH 10, if the separation takes place, if the mixture of amino acid is buffered at pH 10, this side is the positive side of the plate, this side is negative side of the plate. After separation, where these amino acid would be present? To the right and on. Because at pH 10, glycine is what? Negative. At pH 10, ASP is what? Negative, 2 negative. And this is also negative. So at pH 10, all three amino acids in the mixture are negatively charged. So all three are going to move to the positive side. Who will be leading? Who will be ahead of everyone? The one who has the highest charge. So what has the highest charge? 2 minus. So ASP would be here. Who will be behind? Both of them are negative. This one and this one. Then takes the MR. Then the mass. The higher mass, lower speed. So who will be the next then? Then will be the glycine. And the last will be what? Lysine. Okay. Why? Because this is 2 negative. This is 1 negative and this is 1 negative. But lysine is a bigger in mass. Its R group is bigger. So hence it's behind this. Because the rate of movement of amino acid is directly proportional to the charge and inversely proportional to the mass. Okay. So at pH 7, they look like this. When we get separation at pH 7, when we buffer the mixture of the amino acid at pH 7, so we see one of them, for example, the ASP was on this side, glycine was here, and lysine was here, okay, at pH 10. Now let's see how they look like after separation at pH 2. Right? At pH 2. pH 2 pe, look at the pH 2. The glycine is what charge? At pH 2, I'm highlighting it here. What is the charge? Positive. This is the pH 2. What is the charge of ASP? Positive. What is the charge of lysine? 2. All three are the positive. So who will be ahead? 2 positive wala. That is what? Lysine. Okay. That is 2 positive. And that'll be the head. Who will be behind? And who will be behind? Both are positive? No, not ASP. After lysine, both are positive. But glycine is less MR as compared to ASP. So as ASP will be behind. All three move in the direction, but they move like this, right? So here you buffer. Now I hope you understand what is the effect of pH. If pH buffering changes their direction actually, right? Changes their movement actually, right? So at a different pH, once we buffer it, so you see the different results we have obtained, right? Now I'm going to dictate all this and write all this. You have to copy with me what you have to write, okay? So I hope you understood how the separation of amino acid done using the electrophoresis, and this phenomena is known as electrophoresis, in which a glass plate is used, potential difference is applied, and the difference of charge makes them move in the different directions, and similarly their MR plays a role. Okay, so please write down electrophoresis. This technique is known as electrophoresis. Used for the separation of amino acids polyamide gel. There is no apparatus in the syllabus, but I just explaining it too. Polyamide gel applied on a glass plate. Mixture of amino acid. Buffered at specific pH. potential difference applied across the plate. The rate of movement of amino acid. The amino acid move to the respective electrodes. Rate of movement of amino acids is directly proportional charge and inversely proportional to the MR. Molecular mass. Right? And then we discuss the effect of pH. How the pH change turns the amino acids, their nature, their way of movements. Then you have to copy all this. ये कर लिया कॉपी? इसको कॉपी करना होगा बच्चों. You have to copy then all this. This is the effect of pH.
There is delta in your notes group.
Yes.
Will you send these notes to the group?
Yeah, I will.
Just share it now.
Yeah, I'll do it right now. Done? Are you done? Okay, I'm going to share the notes. That's a good idea. We'll stop here. We'll continue this in the next class, how this chromatography helps to separate the amino acids. That's all from my side for today.